My name's Kimberly.
And
we're juniors in AP Bio, and welcome to our Taxonomy Lab tutorial.
Today we're going to walk you through how to analyze taxonomic clades and build a cladogram based on shared derived traits.
The title of our lab is Taxonomic Clades and Cladogram Construction. Organisms that share more derived traits are more closely related on the cladogram.
And the materials you'll need for this lab is
a pen
and your lab paper that you are filling out. And for each taxonomic clade, you're going to write 3 shared derived characteristic traits between each clade. And on the back, you also have a little cheat sheet of common shared characteristic traits used in cladistics and taxonomy. And you'll also need
your noggin.
The purpose of this lab in AP Bio is to explore taxonomy and understand how to create and interpret cladograms, which shows the evolutionary relationships between different species. This is also your friendly reminder of our safety rule, which is to please be careful if the jars in the lab. Do not pick them up because they might fall and break.
So for this lab, you'll be in the lab room and each table is set up with a different clade. In total, there are 8 different clades. And just a reminder, you are writing 3 shared characteristics for each different clade.
For this lab, you're going to be in small groups and you're each going to work together at a station one at a time.
Each group will be in a different station, in a different clade, and you guys will be observing their shared characteristics to put on your paper.
So look at all the different jars and see what is inside the jars and see what they have in common. After the timer runs up, you're going to go to the next station and repeat the process for each taxonomic clade. The first taxonomic clade we're doing is Cnidaria. We have jellyfish
and we have coral. And these are just the few animals that you'll be observing in the clade Cnidaria.
Synamorphies is another word for shared derived characteristics that help define evolutionary relationships between organisms. Some synamorphies for the clade Cnidaria is 1. Nematocyst, 2. Medusa or polyp stages, 3. Diploblastic with mesclia, 4. Epithelioid muscular cells, and 5. Planula larvae. And the next clade we have is Chordata, and this is where you'll find dogfish pup,
a turtle,
a baby crocodile,
a frog. Again, these are just a few examples of the clade Chordata.
Synamorphies for the clade Chordata is 1. Notochord, 2. Single dorsal hollow nerve spinal cord, 3. Pharyngeal pouches / slits, 4. Endostyle, and 5. postnatal. And the next clade we have is Plantae.
One of the examples is basswood.
Another example is larch.
Another one is bean, the flowering plant.
Another one is a lily. And these are just a few examples of Plantae. Synamorphies for the clade Plantae is 1. Chloroplast, 2. Chlorophyll, A-B carotenoids, 3. Storage and starch, polysaccharide, 4. Cellulose cell wall, and 5. Stacked thylakoids.
This clade is Arthropoda.
And one example is a butterfly,
a termite.
Another one is a grasshopper,
a spider.
And these are just a few examples of
Arthropoda.
For Arthropoda, synamorphies is 1. Compound eyes, 2. Jointed limbs associated with the head, biramous trunk limbs, jointed body, and 3. Specialized appendages associated with the head. The next one we have is mollusca. This is where you'll find
chitin,
or a squid,
or a land snail,
or a clam,
or a garden slug.
And these are just a few examples of
mollusca.
Synamorphies for mollusca is 1. The mantle, 2. The foot, 3. The radula, and 4. Pectinate gills.
Okay, this clade is fungi.
And an example of this is a woodland mushroom.
A puffball.
Another one is a bird's nest fungi. And these are just a few examples of
Fungi.
fungi. Synamorphies include 1. Produce cell walls containing chitin. 2. They grow primarily as mycelia or filaments of long cells called hyphae. The next clade we have is porifera,
One of the examples is a marine sponge.
or a finger sponge. And these are just a few examples of
Porifera.
Synamorphies for porphyria is 1. Choncytes. 2. An aquiferous system of external pores. 3. Mineral spicules. And 4. High cellular mobility and totipentancy.
This clade is Echinodermata.
And an example is starfish.
Patrick's cousin, the starfish.
A sand dollar.
A sea urchin.
And these are just a few examples of
Echinodermata.
Echinodermata synamorphies is 1. Calcitic skeleton composed of many ossicles. 2. Water vascular system. 3. Mutable collagenous tissue. And 4. Pentaradial body organization in adults.
After analyzing all the traits and jars, we will use this data to make our own cladogram.
To create your hypothetical cladogram for all the clades of organisms using the evidence you collected from observations made on their shared derived characters. Choose at least one character to be used as a node to separate or connect each clade to the others. Be sure to label your cladogram with the names of the clades and their derived characters.
Now we will give you an example of how to create your own cladogram.
A cladogram is a diagram that shows how different organisms are related through evolution. It's like a family tree but for species. We use cladograms to figure out which groups share a common ancestor and what traits they have in common. To make a cladogram, you must first list the organisms, second, compare their traits, and third, group them based on shared traits from least to most. The more traits two groups share, the closer they'll be on the cladogram. Our cladogram fits because it groups organisms based on shared traits like having tissues, such as in porifera versus the rest, having a nervous system, starting with Cnidaria, bilateral symmetry, such as mollusca and beyond, a body cavity in mollusca, arthropoda, and chordata, a notochord only in chordata, and being deuterostomes in echinodermata and chordata. The final step in this lab will be your whiteboard presentation. You will write the guiding question and your claim on this specific layout. Your evidence will be a photo of your cladogram, and then you will write your justification, explaining how your cladogram best shows the relationship between taxonomic clades. After completing this final step, this will conclude your taxonomy lab. In conclusion, this lab helped us see how evolutionary biology uses evidence, not just appearance, to classify life.
We hope you learn how to identify clades
And build a cladogram.
Thank you for watching our Taxonomy Lab tutorial. Bye!